Welcome students. Today we are going to talk about a very unique perspective of the respiration, which is called beta oxidation of fatty acid. Many a times in the NEET this type of questions comes in the question paper related with the numericals and theory of course. So let us start the beta oxidation of fatty acid. What is beta oxidation? Beta oxidation is a catabolic process by which fatty acid molecules are broken down to generate the acetyl CoA enzyme. This acetyl CoA enters into the citric acid cycle and they produces NADH2 and FADH2, which are the coenzymes used in the electron transport chain. Remember, this breakdown takes place in the cytosol in case of prokaryotes and in mitochondria in case of eukaryotes. Along with that, please note that as a source of energy, RBC use carbohydrates, neuron cells use carbohydrates as well as ketone bodies, and RBC do not have the mitochondria. So therefore, beta oxidation is a catabolic process to break down the fatty acids to generate the acetyl CoA enzymes. See, now fatty acids, well, we'll say about this. Apart from this fatty acid, while amino acids and carbohydrates can oxidize anaerobically, that is without added of the oxygen, fats can oxidize only aerobically. Triglycerols, which is generally called as fats and oils, store the majority of the energy in most of the animals and plants, and these fatty acids of oils contains more double bonds than those of the fats, therefore they can yield more energy. Fatty acid beta oxidation is named so because the beta carbon of the fatty acid undergoes oxidation to a carbonyl group. Why repeat? The beta carbon of the fatty acid undergoes oxidation to a carbonyl group. Remember, free fatty acid cannot penetrate any biological membrane due to their negative charge. So negative charge is important over here. So free fatty acid contains the negative charges. Free fatty acids must cross the cell membrane through specific transport protein. Fatty acid provides very high energy, highly efficient energy storage, which actually delivers more than the carbohydrates like glucose. In tissues with high energy requirement, the tissues that requires very high energy every second, like cardiac tissues or heart, that gets 50 to 70% of energy in the form of ATP productions that comes from the fatty acid beta oxidation. Remember, during the fatty acid beta oxidation, long chain acyl CoA molecules, remember the long chain acyl CoA molecules, which are the main components of the fatty acid, are broken down into acetyl CoA molecules. Fatty acid must be converted to acyl CoA in order to enter to the mitochondria and be oxidized. First thing that we must know. Then fatty acid beta oxidation therefore is a process of breaking down a long chain acyl CoA molecule to acetyl CoA molecules. The number of acetyl CoA produced from the fatty acid that depends upon the carbon length of the fatty acid that is being oxidized. Remember, the carbon length is very important, and from this point of view, always question comes in the NEET level. Let us look at the process of the beta oxidation. We take it an example of 16 carbon chains. Now it is, you see, this is a long chain fatty acid where the beta carbon and alpha carbon is also there. With the help of enzyme thiokinase and cofactor magnesium, coenzyme A, now ATP is converted to AMP, that means 2 ATP is consumed, and this fatty acid is converted to fatty acyl CoA enzyme. fatty acyl CoA. Again, with the acyl CoA dehydrogenase enzyme, it is converted to trans-enoyl CoA, where FAD is converted to FADH2. So we get over here one FADH2, we get over here, where one ATP is actually converted to AMP. So we can say that it consumes 2 ATP, right? 2 ATP. And this process is called as dehydrogenation. So what we will say that the first process, this is called as esterification. Remember this, the first process is called as esterification, and then the second is called as dehydrogenation. We got the FADH2. Now this trans-enoyl CoA enzyme, with the help of the water, you can see, and the enzyme enoyl CoA hydratase, it converted to beta hydroxyacyl CoA, and the process is called is hydration. Now here you will see here the beta carbon, here the 2 hydrogen is added over here, and now slowly the beta carbon which is been active over these fatty acid beta oxidation. Then beta hydroxyacyl CoA is converted to beta ketoacyl CoA with the help of beta hydroxyacyl CoA dehydrogenase. This is also called as dehydrogenation, and we get NADH2 in these conversions. After this conversion, the beta ketoacyl CoA with the help of coenzyme A, it becomes acyl CoA acetyl transferase, and this process now is called as cleavage. What it converted into fatty acyl CoA, which is 14 carbon chain long fatty acids, and then acetyl CoA, which is a 2 carbon long, right? So we know acetyl CoA is a 2 carbon length, and the fatty acyl CoA is a 14 carbon length. This process is called as cleavage. So it starts with esterification and it ends with the cleavage, and these acetyl CoA now enters into the Krebs cycle. Let us look at these numerical point of view of the beta oxidation of fatty acid. First, the formula of calculating energetics for the even chain fatty acid. When we calculate the even chain fatty acid, then the rules are like that. Small n, it is considered as number of carbon atom present in the fatty acid. Then number of acetyl CoA produced will be n divided by 2. Number of cycles for fatty acid will be n by 2 minus 1. And number of reduced coenzyme will be the same, n by 2 minus 1 into FADH2 plus NADH2. This is the formula. Now based on this formula, let us solve one question. Let us take an example of 16 carbon atom fatty acid, which is called as palmitic acid. Very common question over the NEET. It undergoes beta oxidation. Now you see, number of acetyl CoA will be 8, because n is the number of carbon chain. So number of acetyl CoA n by 2, so it becomes n by 2, it is 8 acetyl CoA. Now one acetyl CoA produce 12 ATP in the TCA cycles, that is Krebs cycle. Therefore 8 acetyl CoA will be the 96 ATP. This is the number 1 point. Number 2 point is that number of cycle, how many number of fatty acid cycle over here, we say n by 2 minus 1. So it will be n by 2 minus 1, that is 7 cycles. Now number of reduced coenzyme, it will be 7 into 2 plus 3. Why 2? Because FADH2 produces approximately 2 ATPs and NADH2 produces approximately 3 ATPs, that we generally calculate. So it comes 35 ATP. Therefore total number of ATP will be 96 plus 35 equals to 131. But remember, during the starting of the beta oxidation, in case of esterification, we already utilized 2 ATPs. So the net gain is 131 minus 2, it is 129 ATP. So in that way we can remember, if it is the 16 carbon as carbons, then we can say that it is 8. 8 12s are 96. If it is a 14 carbon, we can say it is a 7. Then 7 into 12, and then 6. 6 into 5, so it is a 30, and then we add it and minus it. So this is the simple way that you can practice. Next, we will calculate the energetics for the odd chain fatty acid. That also comes. 1 year comes with the even chain, and the next year it comes with the odd chain. Remember, in case of acetyl-CoA odd chain beta oxidation, it should be number of acetyl-CoA produced will be the n minus 3 by 2. It is n minus 3 by 2 into 12 ATP in a one short word. N is the number of carbon. Now here n minus 3. Why it is n minus 3? I will come down to it. Because 1 acetyl-CoA 12 ATP, so 12 into n minus 3 by 2. So number of cycles will be similarly n minus 3 by 2. So reduced coenzyme will be n minus 3 by 2 into FADH2 and NADH2. So it will be 2 plus 3, right? Now these remaining 3 carbon, why we minus 3? Because these 3 carbon is the propionyl CoA. It becomes a 3 carbon is a propionyl CoA, which is converted to succinyl CoA, and that enters into the Krebs cycle. Remember, first of all, these 3 carbon within the odd chain fatty acid, the 3 carbon is the propionyl CoA, which is converted to succinyl CoA and then enters into the Krebs cycle. So example, suppose we take 19C fatty acids. So 19C means n minus 3 by 2. Obviously it will be 19 minus 3 by 2 equals to 8. 12 is the number of ATP, so it is 96 ATPs. 1 acetyl-CoA produces 12 ATP in the TCA cycle. Number of cycles will be the 8. So reduced coenzymes will be n minus 3 by 2 into 5. 3 plus 2, 5. So it comes 40 ATP plus 96 ATP. So it is the 136 ATP. Now remember one thing, that this is the thing that it produces. Remaining 3C propionyl CoA, which is converted to succinyl CoA and enters into the Krebs cycle. Now remember one thing, what I have not written over here must be question in your mind. To attract your attention, you must minus 2 ATP from these 136. Remember, this is to attract your attention. I have not written over there. You must remember 136 minus 2 must be subtracted. So it will be 134 ATP. I repeat, it will be 134 ATP. If you like this video, if you feel it is useful, please press the like icon. Please share it to your friends as maximum as possible, and if you are not subscribed my channel, please subscribe it so that immediately you can get such videos immediately, right? Thank you students.